Okay, it started recording. Howdy, today we are going to present Project 5 with the topic humoral immunity. The group mates are Cassie Fernandez, Hannah
Hannah
Garcia,
Garcia,
and Sofia Guerra.
What is humoral immunity? Humoral immunity is a branch of adaptive immunity mediated by B lymphocytes or B cells. B cells recognize specific antigens and are subsequently activated, proliferate, and differentiate. Differentiated B cells can form two types of effector cells or plasma cells, which secrete antibodies or immunoglobulins to neutralize antigens, and memory B cells, which remain in circulation for rapid response to re-exposure and play a key role in long-term immunity. Classes of immunoglobulins. There are 5 types of antibodies or immunoglobulins. They are Y-shaped proteins that recognize unique markers or antigens on pathogens. IgA is found in mucosal secretions such as saliva, tears, and breast milk, and it protects mucosal surfaces. IgD is found in naive B cell surfaces and might play a role in B cell activation. IgE binds to mast cells and basophils, triggers allergic responses and protection against helminths. IgG, the most abundant immunoglobulin in blood, is involved in complement activation, phagocyte response, and neonatal immunity, where it crosses from the placenta. IgM is the first antibody produced in an immune response. It has a pentameric structure that enhances complement activation. The humoral immune response is a 4-phase process. During antigen recognition, the first step of this process, naive B cells with IgM or IgD antibodies recognize antigens on a microbial pathogen, which could be a bacteria, virus, helminth, fungus, etc. With the help of helper T cells or other stimuli, these B cells become activated. They eventually proliferate or multiply to create many cells just like themselves. Finally, in the differentiation phase, the B cells each experience different outcomes. Some B cells differentiate or turn into plasma cells responsible for secreting antibodies. B cells that express IgG differentiate into plasma cells that engage in isotype switching, meaning they can change the class of their antibodies into either IgM, IgG, IgE, or IgA, depending on the specific situation. For B cells that express high-affinity immunoglobulins, plasma cells engaging in affinity maturation results, in which the antibodies increase in their ability to respond specifically to antigens. Lastly, memory B cells, despite not being capable of secreting antibodies, survive for long periods of time circulating the bloodstream and respond rapidly upon reintroduction of their respective antigens.
Stimulation of B lymphocytes by antigen, T-dependent versus T-independent. T-dependent activation, which is most common and provides a stronger response. Follicular B cells with IgM recognize protein antigens, present antigens via MHC molecules to helper T cells. T cells provide cytokines and CD440L signals for strong memory B cell formation. It produces high affinity isotype switching antibodies, which include IgG, IgA, and IgE, whereas T-independent activation is faster and short-lived response. B1 cells and marginal zone B cells recognize polysaccharide antigens. No T cell involvement relies on innate immune signals and produces mainly IgM, low affinity, short-lived plasma cells. Role of innate signals in B cell activation. Complement activation, CD3 and CR2 receptors. Microbial surfaces activate complement system. CD3 complement protein binds to microbes. B cells have CR2 receptors that recognize CD3 enhanced activation. Pathogen-associated molecular patterns, PAMPs, or toll-like receptors, TLRs, Microbial molecules, AMPs, activate B cells through TLRs. An example is bacterial LPS binding to TLR4. Heavy chain isotype class switching. Helper T cells direct antibody class switching via cytokines, interferon gamma, IgG, opsonization and complement activation, IL-4 to IgE, which help with allergen helminth immunity, TGF, goes into IgA and kills immunity, and the process B cells constant reagent of the heavy chain changes, but antigen specificity remains the same. Enables adaptation to different immune needs. Affinity maturation, process that increases the strength of an antigen binding over time. Occurs in germinal centers of lymphoid follicles, driven by somatic hybrid mutation in immunoglobulin genes. Higher affinity B cells are selected. While low affinity ones undergo apoptosis, it ensures stronger immune response upon repeated exposure.
Regulation of humoral immune responses, antibody feedback. It prevents overreaction of B cells. Antibody-antigen complexes bind to Fc receptors on B cells. One receptor recognizes antigens. Fc receptor binds to antibody already attached to antigen. ITMs, immunoreceptor cytosine-based inhibitant motifs, blocking activating signals stop B cell activation. Antibody effector functions. Neutralization. Neutralization blocks microbial invasion and toxic effects. Opsonization enhances phagocytosis by coating microbes with IgG. ADCC, antibody-dependent cellular cytotoxicity. IgG binds infected cells, natural killer cells, to kill the target. Complement activation, classical pathway. IgG and IgM trigger cascade, microbial lysis and inflammation. IgE-mediated defense binds eosinophils, mast cells, and targets parasites and allergens. Role of antibodies in vaccination. Vaccines trigger humoral immunity by exposing the immune system to antigens. Types of vaccines. Mutated, live, weakened microbes. Strongest response, long-term immunity. Subunit vaccines contain antigenic proteins or polysaccharides. Conjugate vaccines, polysaccharide antigens linked to proteins for stronger response. Synthetic vaccines, lab-made proteins stimulate immunity. Viral vector vaccines, used to modify viruses to deliver antigen genes. DNA vaccines, contain plasmid DNA encoding microbial antigens for host expression. Thank you, guys. Thank you for listening to our presentation.